The overall goal of this experiment is to provide an efficient and easily interpretable method to detect the subcellular localization and internalization of expressed GPCR fusion proteins in mammalian cells.
This method can help answer the key questions in the subcellular protein localization field, such as receptor internalization and recycling. The main advantage of this technique is that it can be used easily and efficiently.
Though this method can provide insight into monitoring and locating GPCRs, it can also apply to other systems such as cytoplasmic and nuclear protein.
After recovering and passaging HEK293 cells according to the text protocol, when the cells have grown to near confluence, discard the equilibrated growth medium, add 2 milliliters of PBS, and swirl the dish to wash the cells. Discard the PBS and add 1 milliliter of trypsin EDTA. To completely digest the cells, carefully swirl the dish and aspirate off the solution. Then, put the dish in this 37 degrees Celsius water-jacketed CO2 incubator for 3 minutes. While the cells are incubating, prepare a 6-well plate by adding 2 milliliters of equilibrated growth medium to each well. Then, when the cells have lifted from the bottom of the dish, add 1 milliliter of equilibrated growth medium to the dish. Mix by pipetting and immediately transfer 0.1 milliliters of cell suspension into each well of the prepared 6-well plate. Gently pipette the suspension in the wells and incubate the cells at 37 degrees.